carry out this cycle of growth and division indefinitely. And you don't have to do anything artificial like force them through small pores. It's just spontaneous growth and gentle shaking. Okay, so let me just say a few words briefly about how we think this happens. One thing we realized is when you make these larger vesicles, they have not just a single membrane layer, but usually several. And so when you add food molecules, the first thing to grow is the outermost layer. There's not much volume in between it and the next, and that volume can only increase slowly as molecules cross the membrane. And if that's a slow process, the surface area is growing faster than the volume. Essentially, the only thing that can happen is to extrude or force out that extra surface area as a filament. What we don't really understand is how is the next steps that drive the transformation of the entire vesicle into a long, thin filament. So we're still trying to work out the mechanistic aspects of this transformation. But what we do know is once you've got a long, hollow filament like this, gentle agitation causes it to break up, and then you can grow these and iterate this cycle. Okay, so what started off 6 or 7 years ago as a problem that, it was just baffling. We had not really much of an idea of how to make it work, has turned out in a few simple steps to actually have a very simple and robust solution that works with lots of different kinds of building block molecules, lots of different environmental conditions. And we actually think it's a pretty good model for how primitive cell membranes could grow and divide. Okay, so it's all very nice to have the membrane problem taken care of, but you still need to have genetic information to carry out the transmission of information from generation to generation. And so how could that go on? So this is a problem that has a long history. Leslie Orgel worked on this problem for decades. Almost 20 years ago, he made this statement in a review. I think the problem, the problem is not solved yet. And I think there are a couple of reasons for that. On the one hand, maybe the problem is a little harder even than Leslie thought. On the other hand, there's hardly anybody working on it, so it's maybe not surprising there hasn't been that much progress. So a lot of the work in the area of how early genetic materials might have replicated for decades after the discovery of catalytic RNA focused on RNA-catalyzed RNA replication. And so, for example, Jerry Joyce has made tremendous progress in that area. And we worked in that area also for a long time. For a number of reasons, I've come to think that even though that would have been an early phase in how cells worked, there had to be, before that, some simpler chemistry, a chemically based mechanism for copying genetic information. And so actually most of my lab now is focused on that problem. We're taking a similar strategy to the membrane strategy. What we want now, when our understanding is pretty minimal, is just any system, not a natural, plausible prebiotic system, just any chemical system where we can copy genetic information with reasonable efficiency. And the idea is if we can do that, it'll probably start to give us ideas about how to generate new systems, and maybe ultimately we'll come to something that's reasonable in terms of the origin of life. Okay, so again, let me just show you another one of Janet's little movies just to make it clear what we want. Hey, that's not supposed to happen. Okay, we want to have short little strands, oligonucleotides floating around in solution, together with activated monomers that are just going to diffuse around, find their complementary position on the template, and chemically click into place and gradually build up a complementary strand. And then we imagine some way of separating these strands, perhaps using high temperature, and then repeat that cycle. Okay, so we need a robust chemistry to solve this kind of problem. And a lot of the early work done by Leslie and also by Jerry and other colleagues of Leslie involved looking at this problem from the perspective of RNA. And so I just want to show an experiment of that type. This is actually done by David Horning when he was an undergrad in my lab. These are the kinds of monomers that were developed in Leslie Orgel's lab. So somewhat like a nucleoside, a modern biological nucleotide, except that instead of a triphosphate, we have this phosphate activated with a much better leaving group. So here, a methylimidazole. Okay, so these molecules are relatively easy to prepare. If you make these and feed them to a primer annealed to a template, you can copy part of the template. So we can extend the primer with several Gs if there are Cs on the template. And this is what it looks like. So here's the starting material. Here you've added 1 G over, say, 6 to 10 hours. The second one goes in over a day or so. The third one goes in pretty slowly. So that's one issue. It's pretty slow. More serious issue is that there are 2 potential nucleophiles here. And unless you go to a lot of trouble and tricks, you tend to get linkages through either the 2' hydroxyl or the 3' hydroxyl. This is also something that works pretty well with Gs and Cs, but not so well at all with As and Us. And there are some other issues as well. So we need to get started. To build the system we're trying to build, we need something more efficient than this. Okay, and so this is one of the more advanced systems we've been studying. Again, traces back to early work by Leslie Orgel. But here we've made an additional change. So instead of having 2 hydroxyls, now we have 1 amino group at the 2' position. And so this has some nice potential advantages. Because of the rigid sugar structure, this nucleophile can't reach the electrophile. It can't cyclize, so we take away one of the competing side reactions. Because this is at the 2' position, the polymer that results from polymerization is a 2,5 linked nucleic acid. So it's different from our normal RNA and DNA. But that could have the advantage of making it easier to separate the strands if a duplex is assembled. And this seems to be the reaction that is normally favored in RNA systems. So we thought these were worth studying. And it turned out to be better, I think, than we could have expected. So here are just a couple of experiments. Very simple, just using 1 nucleotide, the best one, activated G. Again, extending a primer where the template is a bunch of Cs. Okay. And so if we just make a primer template complex like this and feed it that activated nucleotide I just showed you, what you get is the primer spontaneously extending without any enzyme. So here's the primer, here are the longer products, and over about half a day, you accumulate very cleanly a full-length product. Okay, so if we had a reaction this good that could copy any sequence, we would be in business. We could just use this as part of an evolving model protocell. Okay, so we don't, and I'll talk more about those problems in a minute, but this was actually good enough for us to do a really important experiment for us, which is to test the idea that this kind of copying chemistry would be compatible with or consistent with our membrane system. Okay, so the experiment is shown over here, where we put this primer template inside the vesicle and then add the nucleotides to the outside. They cross the membrane spontaneously, do the same primer extension, which you see over here. We get the same full-length product. It takes a little bit longer because of the time it takes the monomers to get across the membrane. But it still works. And so it shows that fundamentally these 2 different chemical systems are mutually compatible. So that was very encouraging to us and told us that if we just focused worked hard on the chemical copying part, we could actually combine